Okay, let's wrap up our discussion on the nonspecific or innate side of body defenses, all right? We already talked about the fact that we have these walls around the city, right? These physical barriers called skin and mucous membranes. We have inflammation. If there's a break in that wall, we're going to call for backup. But we also have certain cells of the immune system that I always think of as kind of like security guards that guard the wall. All right. In addition to the properties the wall has, we have phagocytes. Oftentimes, these phagocytes are either stationed on the wall or at the wall, kind of like a security guard would be in one location, just kind of guarding that region. Or we have some that will kind of go in the bloodstream and move around. So think of that almost like casing the perimeter, right? If it's a security guard that does that, all right? So we have these physical barriers and inflammation. Let's now talk about some of those cellular barriers, the phagocytes. So here we have actually an image right down here in the bottom left of a phagocyte, okay? And you can see an ingested bacterium right inside that cell. And, I'm going to show you some videos here in a second, but obviously these phagocytes typically are going to be much bigger than a single bacterium. These are animalian cells, right, first of all. Macrophages, just by the very nature of the name itself, macro tells you they're big, right? They're big cells and they're big eating cells, right? Phage for eating. Okay, so these are big eaters. Monocytes develop into macrophages. Macrophages are one of two major phagocytes I'd say you definitely want to know for me, okay? Phagocytes come in many forms, but neutrophils would be the most abundant, so there's our second, and macrophages are also very active in the phagocyte process. Okay, so here we also see a phagocyte and two cocci organisms, it looks to be, right? Now we know these kinds of things from what we've learned in micro. These two cocci organisms clearly are now inside the cell. Let me show you, this is an image of what these macrophages can do. These macrophages can really morph their membranes, all right? So you can see this blue cell here really has reached out with the different pseudopods, kind of like they're almost like feet, right? Pods for feet. And these are able to actually not just grab onto surfaces, but they're able to grab onto bacteria like these bacillus organisms I see in this picture, all right? Let me show you a video here. This is a video, pretty cool. It's just crazy to see these in action. But you can see here that there's some cocci organisms around, right? Here's a cocci organism, here's one here. And that these can be grabbed onto by this phagocyte here in the middle, right? And they can be pulled in and destroyed. There are lysosomes inside the phagocyte, and those lysosomes will help us to destroy. Oh, caught one right there, right? Okay, so that will be soon brought into the cell and destroyed by those digestive enzymes. Here we have another video showing the phagocytosis of a bacillus type organism, Shigella. All right. Now this is also pretty cool because it catches one of them here, right? But these two guys have a getaway, right? They actually move away from the phagocyte. And of course, we know they have flagella, right? And they're capable of moving towards and away from things, all right? Now, this particular little image here, this shows you these blue compartments. These are all lower pH compartments that are the lysosomes, all right? And the lysosomes, those enzymes inside those lysosomes usually work in a little bit more acidic environment, around a pH of 5. So what they've done with this cell, if they stained it with a pH indicator, this stains blue when it's a little more acidic. And that kind of really lights up those lysosomes inside this phagocyte. So, two biggies you need to know. Neutrophils. And for your information, neutrophils respond quickly. They are some of the first cells on the scene when inflammation occurs. So, I think it's like good 60 or 70% of the white blood cells in the bloodstream are going to be neutrophils. If you find a tissue and you've done kind of a sampling of that tissue, you look at it in the laboratory and you see a lot of neutrophils, you know it's a recent event. Also, monocytes, these respond more slowly. And if you look at a tissue sample that's been collected and you see a lot of monocytes that have developed into macrophages, well, they're probably been there for a while. That damage has occurred and it's been there for a while, right? Because these are slower responders. Now, some of these like I say, some of these macrophages are almost like security guards, right? And they stand their post. They may be found in a particular organ, and we say they're fixed macrophages. Some of these you might recognize maybe from previous bio classes. Langerhans cells, they specialize in the skin. These microglia, well, glial cells are brain cells. They're not the nerve cells, but they're more abundant than the neurons. Kupffer cells are in the liver, and dendritic cells are in a lot of connective tissues, like the connective tissues underneath your skin, for example. All right, areolar, irregular, dense connective tissues. I'm not going to get into those details with you again in this course, but those are the places you'll find those dendritic cells. Now, like I said, there are other phagocytes. They specialize depending on the organ system and the pathogen then involved. All right, but the neutrophils and monocytes, definitely one of those. You'd also want to know generally the steps of phagocytosis. So once a phagocyte has encountered some pathogen, perhaps a bacterium like you see in this image, then it has to have certain steps, undergo certain steps to go to destroy that particular cell. And they're kind of common sense, what would happen here. It has to attach to the cell, adherence, attachment, something like that, right? It has to engulf, and this would be more or less an endocytosis process where it takes in whatever it is, a bacterium, let's say. It's going to fuse that little vesicle that we have with the lysosome where the enzymes are. So we have lysosome fusion. There's the digestion of whatever that thing is, in this case, this blue bacterium, and then exocytosis spits the stuff right out, right? So basic steps of phagocytosis are kind of intuitive, right? Attach, bring the thing in, stuff it in the recycle center, right? That lysosome, digest, and spit out through exocytosis. Now I want to just mention here real quickly, because a lot of times this is kind of confusing to people. Why wouldn't a macrophage just eat up any cell? Eat up our own cells, right? Because they do eat up dead cells, right? So how do they know the difference? It just so happens, at least with respect to pathogens, that these phagocytes are a little smarter than we give them credit for. I'm saying this is a non-specific response, right? These phagocytes are in the innate or non-specific section of this unit, but they do possess what are called pathogen recognition receptors. Some reports will call they're like toll-like receptors. They call them toll-like receptors, but these are found on phagocytes, and they give them the ability to recognize the pathogen, just like the name implies. It's abbreviated PRR. And it just so happens that pathogens exhibit certain molecular patterns on their surface that these PRRs can recognize. Okay? So the pathogens coming in with what are called pathogen-associated molecular patterns. Now, these patterns are pretty cool, and it gets really complicated very quickly, but I just want you to know these are not just physical patterns. Pathogens will exhibit a time-wise set of steps when they infect, when they are virulent and cause disease. So if you compared E. coli that is harmless in our system, or some other bacterium that's harmless in our system, it lives on our mucous membranes in the respiratory tract or in the digestive tract. It lives on those membranes and is associated with those tissues all the time without any harm. And then you look at a pathogenic version of that species, same species, but it's a strain that causes disease, you would find that the molecular patterns would be different on the disease-causing virulent form of that species. And these phagocytes can recognize that difference in patterns. All right? So normal flora, in other words, do not exhibit the same PAMPs, P-A-M-Ps, as their nasty cousins that do cause disease. Now, I think in your coronavirus videos for that project, I think the guy referred to these as damage-associated molecular patterns, and that's pretty much the same thing. All right? Okay, finally, last thing to talk about when it comes to nonspecific is fever. Fever is a part of the nonspecific response, and it so happens that leukocytes, white blood cells, when they become more active, do produce something called pyrogens. These pyrogens, if you think about it, pyrogens, the pyro part makes perfect sense, right? Pyromaniacs start fires, all right? Gen is you're generating something, right? You're making something. So these start a fire. These chemicals travel to the hypothalamus. The hypothalamus happens to be where we set our, it's like our thermostat, where we set our temperature for the body to exist at. And this can turn up that thermostat. Heat's not a bad thing, right? A mild fever is actually not too bad of a situation. It's going to enhance phagocyte activity, immune action in the damaged regions, but and increase tissue repair. But it is going to kind of put a damper on the pathogens activity. Again, a good thing. But when this high fever is, when you get a high fever, we're talking much higher than a mild fever, this can damage all proteins. So we worry about a high fever. Mild fever, not too bad a deal. It's going to actually help us. All right, so I'll leave it at that for the non-specific immune system. 2 clips on that and we are done. All right, next time we're going to launch right into the specific or adaptive immune system and get into the real nitty-gritty there with the antibodies and the kind of the cool stuff where we build an immunity. All right.